Lesson 8, Principles of Inheritance and Variation. In today's video, you are going to learn about a new concept about the post-Mendelian inheritance pattern, or as a whole the inheritance pattern in the living world, and this is called as linkage. But before discussing about what is linkage, let's have a quick revision of the last class. In the last class, it was discussed about the chromosomal theory of inheritance. Chromosomes, they are the physical basis of inheritance pattern, where the genes, or this is the part of the DNA that expresses a particular character that goes from one generation into the next. And in that, we discussed some salient features about the chromosome as chromosomal theory of inheritance, postulated by Walter Sutton and Theodore Boveri in the year 1902. In that, you learned that the chromosomes, they are responsible for transferring characters in the form of genes from one generation to the next. That means we can say that it is a bridge between one generation and the next, and that takes place through the formation of the gametes in both the male and the female parent. So this was a small summary of what was learned in the last class through the chromosomal theory of inheritance. Now let's come to the phenomenon that is very much responsible for the transmission of characters from one generation into the other, and this will put a new dimension to the whole pattern of inheritance in any kind of living organism. What is linkage? The phenomenon in which linked genes tend to go together into the same gamete through generation after generation is called linkage. And the term linkage, it was coined by T.H. Morgan in the year 1910. Morgan, he was working on Drosophila melanogaster for studying the inheritance pattern of the different characters. As should be the Drosophila, which is a fly, or that in general terms we call it as the fruit fly. They also bears many contrasting characters. So while studying the inheritance pattern in Drosophila melanogaster, Morgan, he came across some new facts about the whole inheritance pattern. And in that, he coined the term linkage, and the definition of linkage means that the link genes, they always remains together generation after generation. That means that these link genes in the chromosome, they always goes to the same gamete and they never segregate from each other, or they never get separated from each other. So this tendency of the genes is only called as linkage. Now let's try to learn this with the help of a diagram. As you know now that T.H. Morgan had taken Drosophila melanogaster for his experiment on inheritance. In Drosophila melanogaster, there are 4 chromosomes. And out of these 4 chromosomes, one is the sex chromosome, that is chromosome 1 that is given there in the diagram. So that is the sex chromosome, and the rest of the 3, that is chromosome 2, 3 and 4, they are the body chromosomes or autosomes. In that, the diploid chromosome number will be 8 and the haploid chromosome number, that will be 4. Now, as you can see in the diagram that there are some characters that is written with the chromosomes here. As for example, let's take the chromosome 2. So there in that chromosome, you can see that there are 10 genes, and those 10 genes are the genes that will code for the character of the antenna, and here it is Aristotle's antenna. Similarly for the characteristics of wings, and here this chromosome 2, it will code for the second gene, it will code for the curly wings. The third gene, it will code for the clotted eyes. The fourth for the daughterless character. Fifth for the black color of the body. Then sixth, it will be for the purple eyes, etc. Now, these genes which are present on the same chromosome, they will be called as the linked genes. And as you can see there, that it maintains a particular distance. That means all the genes, they are not together. They are present over a distance. Some genes, they are nearby each other, and some genes, they are distant from each other. But as they are present on the same chromosome, that is why they are called as the linked genes. And according to the concept of linkage, it was said that these linked genes, they will pass from one generation to the other during the time of gamete formation together, or they will always tend to remain together generation after generation. From the diagram, as you can see that each gene, it has a definite position in the chromosome, and that position, it is called as the gene locus. So every gene, it has a definite locus in the chromosome, and they have a particular distance in between them, or in between the link genes. And this distance between the link genes, it determines the degree of the linkage strength, or how strongly they are linked with each other. And it is told that closer the distance, more is the linkage, and widely located the link genes are, they show weak linkage. That means more the distance, the linkage or the strength between these genes, it will be weaker than those genes which are closely placed together, or those genes which are nearby each other. So the distance between two linked genes, it is important to maintain the phenomenon that whether a particular gene, it will remain in the same chromosome generation after generation or not. So this is a very important fact about the whole linkage pattern. Linkage, it can be represented with the help of a cross. Now let's take this example for the cross. Here the parents, they will bear two characters. One, that is the shape of the wing and the other one, that is the shape of the abdomen. Here one parent it will be dominant and the another it will be recessive. So here let's consider the male parent to be with the characters long wing and the broad abdomen, and the female parent with the characters vestigial wing and the narrow abdomen. That is on the sides of their wing and the abdomen along with the shape. Now this long wing broad abdomen parent, they will bear gametes with long wing and broad abdomen genes, and the female parent, they will bear the gametes with the vestigial wing and the narrow abdomen genes. And these genes, they are the link genes, as they are present on the same chromosome. That means the long wing and the broad abdomen, they won't be separated and they always tend to remain together generation after generation. So similar is the case with the vestigial wing and the narrow abdomen, that they are present on the same chromosome, as discussed earlier in the diagram, that they will present in the same chromosome, and these are the link genes. Long wing and broad abdomen genes, these are the link genes in the male parent, and the vestigial wing and the narrow abdomen wing genes, they are the link genes in the female parent. Now as they have produced the gametes, the male, it will, as we know that it will be at the male gamete, and the female individual, it will be at a female gamete. Now when the male and the female gamete, they fuses with each other, now this is the new combination. That means we get the heterozygous individuals now. Here the maternal and the paternal chromosomes, they are present in that particular resultant individual in the F1 generation. The new individual or their offspring of this cross, now it will be a two types of chromosome, the paternal chromosome with long wing and broad abdomen character, and the maternal chromosome, it will have the vestigial wing and the narrow abdomen character. So I hope that till here it is quite understandable that the gametes, they have fused with each other. Now this new individual or their offspring, now they bears these two chromosomes in the same individual, or they are the heterozygous individuals now. And because of the presence of the dominant genes, that is the long wing and the broad abdomen, here all the offsprings or the offspring, it will be a long wing and the broad abdomen only as the phenotype. Now what T.H. Morgan had done next, he self-crossed the F1 generation. Now while self-crossing the F1 generation, he got the resultant ratio in the F2 generation as 3:1. So that is the dihybrid phenotypic ratio. Why it is called as dihybrid? Because here two contrasting characters are taken. That is the shape of the wing or the size of the wing and the shape of the abdomen. And here three individuals will bear long wing and the broad abdomen characters and the vestigial, and one individual it will bear the vestigial wing and the narrow abdomen character. Now let's come to the point here. According to this phenomenon, it is said that the link genes, they tend to go together generation into the same gamete generation after generation. So that was the definition, or that is the concept that was given by T.H. Morgan. But what you can see here, that is a dihybrid cross. Why dihybrid cross? Because here two contrasting characters are there. And according to Mendel, now again we have to go back to the principles of inheritance that was proposed by Mendel. And in his third law, he said that these genes, they follow independent assortment. And because of this independent assortment, Mendel got 4 types of gametes for the sweet pea. But here in the F1 generation, individuals, in case of linkage, which was proposed by T.H. Morgan, he again got 2 types of gametes. 50% of the gametes they bear long wing and the broad abdomen genes, and 50% of the gametes they bear vestigial wing and the narrow abdomen genes in the F1 generation. So here there is a difference of opinion between these two scientists, that is Mendel and T.H. Morgan. But Mendel was also not wrong, and T.H. Morgan, he was also not wrong. In case of Drosophila, as we know now that the characters for long wing and the broad abdomen, it is present on the same chromosome. Therefore, these 2 linked genes, they will tend to go together in the same gamete. Similarly, the genes for vestigial wing and the narrow abdomen in the F1 generation, it will tend to go together in the same gamete. As the genes, they will not separate, only the chromosomes that is going to separate. So here we get only 2 types of gametes, and as this is a dihybrid cross, again though it is a dihybrid cross, here only 2 types of gametes are found, and these 2 types of gametes, when they fuse in the next generation during self-crossing, we get 3:1 ratio. 3 long wing broad abdomen individual and 1 vestigial wing narrow abdomen individual. Now in order to test it, again T.H. Morgan performed the test cross. Now what is a test cross? When a cross is done with the F1 hybrid with again the recessive parent. Here the recessive parent, this is the vestigial wing, and the narrow abdomen parent, that is the recessive parent. So when T.H. Morgan crossed the F1 generation individual, that means capital V, capital A, v, small a individual with again the recessive parent, that is small v, small a individual, he got the test cross ratio of 1:1. So this is the result of the test cross ratio. And this result, it was quite different from the Mendel's result. Though both these 2 scientists, they had done the cross taking the dihybrid cross only. Now let's discuss about the types of linkage. There are 2 types of linkage, that is complete linkage and incomplete linkage. Now what is complete linkage? Complete linkage, it was again termed by Morgan in 1919, and it is when the linked genes or the genes located on the same chromosome do not separate and are inherited together over the generations due to the absence of crossing over. Then it is called as complete linkage. So there is another phenomenon that happens parallelly along with linkage. As we know that meiosis, cell division, takes place during the time of formation of the gamete, and during meiosis cell division a very important mechanism takes place, and that is called as crossing over, where the chromosomal arms, they gets exchanged. In that When the genes, they are not separated by crossing over, then only we find complete linkage in our nature, and this is quite rare in the nature, because beforehand we know that the strength of a linkage that can be affected due to the distance between the two linked genes. So as we know that when the distance that is more between two linked genes, so there are chances of weak linkages, and in that case crossing over can take place. And whenever the distance is less, so there is no chance of crossing over, and such chances are quite rare. So therefore complete linkage, it is quite rare in nature. Now let's come to incomplete linkage. So when genes present in the same chromosome have a tendency to separate due to crossing over, and hence produce recombinant progeny besides the parental types, then it is called as incomplete linkage. And in that case the linkage ratio that may vary. As for example, according to one of the cross performed by T.H. Morgan, so there he got the linkage ratio to be 9:1:1:8. So this is the phenomenon that is called as linkage and the types of linkage. Now there is another term which is called as linkage group. So a linkage group is a linearly arranged group of linked genes which are normally inherited together except for the crossing over mechanism. Therefore, the number of chromosomes, or that is the haploid set of chromosomes present in a nucleus or present in a cell, so that determines the linkage group. As for example, in case of Drosophila melanogaster, there are 4 linkage group because there are 4 pairs of chromosomes. In case of human beings, as we know that 46 total chromosomes are there in the diploid set, and therefore the linkage groups will be 23. That means the haploid set. Say for example, in case of pea plant also, so there are 7 linkage groups, and the diploid set for the sweet pea plant, it will be 14. So these are some terms that we discussed in today's class. So in the next class, I'm going to discuss about the term crossing over in more broader sense. Thank you.